Hi, my name is Vivek Mutalik. I'm a scientist at Physical Biosciences Division at Lawrence Berkeley National Lab at Berkeley, California. Today I'm going to talk to you about technical challenges in the synthetic biology field. So let's begin by defining the synthetic biology. As a field, it is a design and construction of new biological systems and parts, and also redesigning the existing biological systems for useful purposes. Here the main idea is to use computer-assisted design approaches and engineering approaches with biological research. Essentially, engineering biological functions, making them easier, efficient, reliable, predictable, and safe. As a field, synthetic biology has promised a lot of solutions for a variety of problems that we are having today. Either it is in energy, environment, agriculture, health, chemicals, food, or also the understanding biological complexity. Synthetic biology has promised solutions. As a field, synthetic biology has applications in variety of industries, whether it is energy, environment, and agriculture, health, and chemicals. As you can see, there are a variety of applications, and each of them have their unique challenges to overcome. Today I'm going to talk to you about a few of these challenges which can be generalizable across different applications. So researchers have developed a variety of approaches to take a gene cluster from one organism, put it in across different organisms to express it and produce whatever the chemical they are interested in. They've also designed and executed different genetic circuits. Variety of pathways have been built over the decades to produce particular interesting chemicals. In this case, it is artemisinin production, which is anti-malarial drug. Or as you know, there is a iGEM competition, which is a worldwide phenomena where there are undergraduates from a variety of different countries participate, where they come up with innovative ideas, and using synthetic biology, they solve different solutions, different problems. Now, over the years, people have used variety of approaches where they have developed sophisticated genetic circuits, whereas this is a dynamic feedback regulation where particular substrate is producing the metabolite, which is a toxic to the cell, producing the product. So metabolite can be used for giving the feedback to the upstream pathway. It represses the upstream pathway and activates the downstream pathway, thereby reducing the toxicity. Also, in Drew and Dee's lab, they have refactored bacteriophage T7 with the 12 kb replaced with the engineered one. From Chris White lab, there is a whole refactoring of a nitrogenase cluster where they have developed more than 500 gene clusters going through design, build, and test and learn cycle. Along with that, we have already seen there is a whole synthetic chromosome synthesized in yeast. And also, as you can see from here, there is a Craig Venter Institute, obviously, done a 1 megabase DNA from cellular DNA from a synthetic approaches. Other than these special cases, other than that, over the state of the art has been slow, expensive, and unpredictable. Usually, the way this system has been working is PhD level scientists plan and execute the material, and they test them and adapt them and manually assemble them. Usually has an ad hoc approach where the application is defined and then they couple design and fabrications and go through the iterative cycle, and usually we don't keep track of the mistakes made or the errors done. So we don't know what are the lessons that we learn from every cycle. So if you can see, if you zoom out and see whether every application has its unique challenges, and it needs a lot of money, time, and labor to optimize because it's not a simple solution. So in this case, a bioprocess platform, you can see that it has got multiple branches. It has got substrate needs to be engineered, product needs to be optimized, recovery of products, and then also reaction systems, conditions need to be optimized, and so on and so forth. So there's a multifactorial problem we need to overcome, and gene expression is one of the simple challenges that is involved in this process. So if we give you a simple test to solve with the state of the art methodology that we have today, take your time to calculate how much money, time, and labor is needed to doing all of these simple projects. Even though they look simple, they have a whole challenges of making them efficient and selective. So for example, can you engineer a pathway that yields 200 grams of isobutanol in a model organism in shake flask? What if I want to refactor a gene cluster identified in organism X and express it in a model system or model host which produces an anti-cancer compound Y? And the third challenge would be, can you engineer a phage which can diagnose the pathogenic microbe which is a drug resistant? And then finally, what efforts we need to do to design a bacterial system or virus for cancer treatment? And the answer is, we are not really sure how much money, time and labor is needed to do all of these things. And people have realized already that the challenge is not only the DNA synthesis, which is getting cheaper and cheaper, though we have a capability increased, our ability to design and put this thing together, we haven't really gotten smart at that. So right now still we have ad hoc genetic engineering, as I mentioned. We have limited well characterized parts, and there is no reliability and predictability associated with the parts. We don't know how to put these things together such that they retain their functionality. And then finally, also when we put these parts together into circuits, we are not really sure what are the emergent properties come up with these complex systems. And then context issue matters, where the genetic circuit is, what is the cellular component, what is the next region of the DNA. And what happens is finally, we are not really learning anything from our failures. We don't keep track of the failures and we don't know what is happening. And then finally, as you know, that we don't know enough of biology really to make the synthetic biology predictably engineering discipline. So how do we really accept all of these challenges and leap ahead? What do we need to do? So as a field, people have already proposed many of these things. This is a Drew and Dee's very famous review in Nature, which particularly shows three steps we need to do so that we can start solving genetic engineering to the next level. So for example, we need to decouple the design and fabrication. We need to standardize parts and part junctions, and we need to abstract a genetic function. So abstraction in terms of whether it is a DNA or it can be a part or devices or systems, we didn't need to know everything to the detail of DNA sequence. We can abstract to a level where we are working and simplify the problem. So if you see in a simple way, if I define an application, the question should be, how do I design it? What specifications are necessary? How do I model it in a computer such that I know what I'm designing, and then choosing the parts and then building them. After that, we characterize it and we debug. And based on that, we can actually find out what step we need to debug. As you can see, the design, build, test cycle that I'm mentioning here is pretty much same for all of the things. Either it can be a software for synthetic biology, or it can be a host system. If I want to engineer a host system, what I need to do. And then how do I design and build, test, learn cycle for a pathway or a device system that we want to engineer. Or it can be a DNA part. If I'm engineering a promoter, I need to know really how do I design it, how do I build it and test and learn. So this cycle of iteration applies to all of the branches of synthetic biology. So if I give a quick primer on regulation of gene expression before we go ahead. Here, RNA polymerase binds to promoter in E. coli, and then it produces all of these three, it's a polycistronic mRNA, it produces this mRNA. Once the mRNA is produced, ribosome binds to the ribosome binding site, and it produces these proteins. As you can see, each of these processes are multi-step processes. So for example, in transcription, we have initiation, elongation, and termination. And all of these three things are regulated by multiple different motifs. Same as for translation, it goes through multiple steps: initiation, elongation, and termination. And each of these steps are regulated by multiple different factors. So as you can see, if I want to optimize the protein production, I need to worry about all of these factors along with mRNA degradation. And so you can see that if I want to really turn the knobs on gene expression, I have a variety of solutions I can approach. So I can change the copy number of the DNA. I can use promoters, sigma factors, RNA polymerases, and so on. I can use variety of these new knobs such that I can tune up the protein expression. So as a protein expression is a huge complex system, protein expression is regulated pretty much by everything in the cell. So it can be a whole transcription machinery along with the translation machinery, all regulated protein expression. It also get impacted by the conditions, replication of the cell of the DNA, and also the growth rate, degradation of mRNA and proteins, folding factors, and so on. So how do we really approach and improve the functional composition within all of this coupling and complexity of varying cellular contexts? So this is a slide from Adam Erkin et al. Where the interesting idea would be, if I take this expression unit or a cassette where I'm producing a protein, and if I put it across different sites on the chromosome, how does it impact the gene expression? How does it impact if I take this same gene expression, put it on a single copy vector or a multi-copy vector? And what happens if same things get impacted by the environmental context? Whether I grow cells in a test tube or I grow them in a shake flask, how does it impact in a reactor? If I put the same expression unit in a different strains, how does it impact the gene expression? So how do we take care of this all context reliability matters into when we are measuring a subunit? So there are early methods where we started to assemble parts. This is a registry of standard biological parts which started to catalog parts from iGEM teams. This is a Joint Bioenergy Institute data sheet where they have a plasmid library and they characterize in different conditions. This is a SEVA library from the European group where they have library of vectors. And this is the data sheet from Drew and his lab where they have shown how to really generate a data sheet of parts under different conditions, such that when we read this data sheet, we will know what happens with the part, how does it behave in different contexts. But now there are a variety of approaches people have put together. There are ligation independent methods, restriction site independent methods. But let's imagine if you put together all these into a different circuit, there are different failure modes that can happen and which impact the performance. So for example, now we know that promoter context is very important. It can impact the protein expression by changing the promoter strength. So we need to put an insulator, but we don't know what is the insulator region. We don't know what is the RBS context, what needs to be around RBS. What happens in between these junctions between these parts? And what happens also the scar issues, if there are restriction enzyme scar between these parts, how do they impact the overall gene expression? We also need to worry about crosstalks if there are some parts which interact, which should not interact. And then also we need to worry about input-output dynamic range of between different gates and then transcriptional read-throughs. So maybe the RNA polymerase does not terminate at the terminator, it reads and interferes with the downstream promoter. And then finally, we need to also understand how do we generate diversity between the genetic circuits, and if we generate diversity, how do we really screen them or measure them? And then finally, how do we take all of this big chunk of DNA in design structures, how do we put them in a particular chromosomal or genomic context? And then how do we really quantify by using in silico methods? How do we model them? How do we approach these solutions? So one of the approach people have come up, particularly in BioFab, where we have a promoter and then downstream UTR and then driving the gene expression of a gene of interest. And these particular functional units, expression unit, are insulated by two different insulator regions. One is upstream insulator and the downstream insulator. So the upstream insulator is made up of a terminator and some regions which we call insulators, and the downstream is another terminator. So the idea is that there is no traffic going inside the transcription unit, and nothing is coming out of this unit. So there are a variety of many publications have come up in recent years where people have really characterized the parts and the junctions. So in this case, there's a promoter library. Variety of methods have been used to generate promoters for E. coli. There are terminators, library of terminators which are available for E. coli. And also there are junctions. So particularly in this case, Chris White's lab has published this paper where we have a ribozyme between a promoter and then the UTR, where it can cut down, restrict the mRNA such that there is no interference between these parts. Also, BioFab has shown this bicistronic design where there is a upstream RBS and then the downstream RBS and then the small peptide coding between the upstream region of interest, where if there is any hairpin formation between the RBS and the gene of interest will be removed by the helicase activity of the ribosomes. So it has shown that using these approaches, if you have a different genes, let's say in this case GFP and RFP, you get a pretty linear dependency. There's a rank order is maintained across different contexts. Also, there is a study here shown on the extreme right, where there is a 5' 3' UTR engineering has been done such that there is no dependency between these parts. And then the most important also unanswered question here is the optimal design conundrum, where we don't know what are the really, how do we codon optimize if we are expressing a gene from different organism in E. coli or any other bug, for example. So we need to really spend more time and investment to understand how does the gene design is happening and what is the optimum solution. There are some biophysical methods. For example, in this case, RBS calculator from Sally's lab has shown that this RBS calculator can be used where biophysical methods are used for expression optimization in a bigger circuits. So one of the things that we always not really take care of is a host and environmental context. So let's imagine this is my gene of, this is my construct that I want to really producing whatever I want to produce or doing the program that I want to engineer. We forget is that it's in the context of a whole cellular genetic material, genetic program that already it is evolved into. Then also we forget that is the metabolic circuits, which is part of, so when we take about the context and host context, particularly we don't take care of these issues. So what happens with that is, because of these dependencies, this circuit might be using gene expression machinery from the cell. It might be titrating away the protein degradation machinery. It is using too much metabolites, cofactors impacted by the replication machinery and so on. And because of that, cell surfaces, it exhibits the burden on the cellular physiology. And because of the burden, cell goes through this toxicity response or stress response, and it impacts the growth rate, and then the circuit performance is impacted. So there are a variety of methods people have come up with. One is, but I'm going to show you two different methods. One is the relative promoter units from Drew and his lab, where they have taken these two different promoters, they sent across different labs and they tested the performance of those. As you can see here, the one promoter white promoter, it shows a different performance across the labs, and the same thing in the second gray bars are showing a different promoters. As you can see, each same promoter shows a different performance under different same conditions in a different labs. Obviously, how do we nearly normalize this? So one idea is to use a relative promoter units where they are dividing one of the activity across, we assume the another promoter activity, by reducing the context variability in a way. And then the second solution is come up with a part score. How do we really score a part across different conditions? And this is the work from BioFab, where we took 7 different promoters and 11 different UTRs driving GFP or RFP on a medium copy plasmid. So when we measure the fluorescence of these two proteins, we see that we can only explain about 40% of the data. So if we use a simple factorial ANOVA, we can come up and score these parts across different conditions. So I'm showing here part score across different promoters and UTRs, and the error bars are showing how does a particular part varies with respect to its upstream genetic context or downstream genetic context. So you can imagine having this kind of a database with the part scores helps in designing and choosing parts for which are appropriate, which are reliable for your application across different systems. So the second thing is, how do we really debug the failure modes that we are having with the host engineering? So there are a variety of methods people have used, and there are a lot of genetic approaches, genetic engineering approaches currently available. And some of them are shown here. MAGE, Tremor, and Cas9 systems have been really useful in E. coli, and not only E. coli, Cas9 has been shown across different systems. So in this case, what happens is you can create mutations or insert a particular gene, and you can have multiple levels of genome engineering scales. And by that, you can reduce the toxicity and effects on the growth rate and physiology. So when there are also other methods, you can also quantify. How do we really quantify the stress levels? And there are methods people have used by not only quantifying but also using a feedback regulation across the circuits. So you can sense the stress, and then you can really feed back into your circuit such that it performs with respect to its stress. It should not impact the cellular physiology. So one of the examples is from Kiesling group, where they have shown that acetyl-CoA can be converted to mevalonate and converts into FPP, which is toxic to the cell. and FPP goes into amorphidine. So you can imagine they found out that if you can remove FPP from the larger pool, you can reduce the stress levels. So they found out FPP responsive promoters, and they use that as down regulating the upstream promoter of the pathway and activating the downstream promoter. By that way, you can remove the FPP from a toxicity level to the much better physiologically relevant levels of the cell. Then the second one is from Tom Ellis' lab. What they did was they have a stress detection methodology where they have a GFP cloned on the chromosome, which can quantify the stress levels of the protein that you want to express. By quantifying that, you can find solution how do you remediate or debug the stress levels. So as we move into bioeconomy, researchers have started thinking about how do we contain the engineered organisms when we released into the environment. So biocontainment is one of the major challenges research community is thinking about. How do we really safeguard, and the variety of strategies people have used over the years. One of them is engineered exotrophy. In this case, toxicity of that particular system can be engineered. When it releases into the cell doesn't survive because of the toxic gene expression. The second method is you have deletion of an essential gene, and you supply an amino acid, and the cell will survive in supplying in the container inside the contained environment. But when it released into the cell outside environment, it doesn't get these essential amino acids, and the cell doesn't survive. And then the third one is the induced lethality, where what happens is when particular cell gets released into the environment in presence of an inducer, you have a gene expression which is toxic to the cell, and cell survival compromises. And then finally, gene flow prevention, when particular plasmid is you have an environment and it gets released into the cell or transfers into the different organism, the toxicity of that gene expression will kill the cells. There are very new methods come up which are multiplexing. These are recent papers. This is from a Church lab, and particularly they recoded the E. coli such that all the UAG codons are now UAA, and now this recoded organism, E. coli, can researchers can engineer all the essential genes to have UAG as a codon for a synthetic amino acid incorporation. So what happens is you need cells to survive, it needs this synthetic amino acid to be supplied. So in absence of this in the natural environment, cell doesn't survive, and there are very few escape mutants because of this mutation. This UAG incorporation has been done on multiple essential genes. Then the second approach recently has come up with where they have a essential histone genes. These two genes are regulated by PGAL promoters, which are activated by JEV protein in presence of an inducer. So in absence of inducer, these two essential proteins are not formed, and the cell doesn't survive in the natural environment. And the second level of redundancy they have incorporated is they have induction of a CRE will cause the recombination between the LOXP sites, and then you lose one of the genes, and the cell is incapable of surviving in the natural environment. So if you think about it, we need all of the different tools along with wet lab tools in the lab. Also, we need a software methodology to handle all of the different challenges that we have. So defining how do we design it and what are the tools to design, we need softwares. We need methodology to model different approaches and applications. We need a software, and along the way we need different software platforms. So I have listed here some of them. For example, we need design and statistical approaches. How do we use design of experiments methodologies? We need how do we store metadata? How do we store part data? And then the sample tracking and strain location and so on and so forth. There is a lot of open applications for software development for synthetic biology. So this is shown in one of these reviews where, let's imagine I have an application definition, we have a specification. Based on that, we can design the parts, design the system, we can define them using the equations. We can solve them and predict on the computer how does it behave. Based on that, we can choose parts using different databases that we have, define them and to convert them into these sequences, and then assemble them in silico and go into the wet lab and build them in reality, or maybe order it through the synthesis companies and then check the experimental data with the in silico predictions. There are a variety of software people have been developing, but there is no one-stop shop for all of these tools together. We are still limited by how do we really keep the samples tracking, what are the failures, how do we keep track of the failures. So we need a lot of improvements. How do we handle the data? We also need move away from simple data tables. So we have been all the researchers still use Excel sheets to check the data or analyze the sequences, analyze the data sets that they are having. We need to move away from that to more design and sequence constructions where design is explained and we have the data along with the designs. We also need more visualization. How do we really access the data and visualize the data? How users are going to use it, score it, or there are more user friendly and a nice experience for using the data. So also if you zoom out from all of these, there are multiple registries coming up across the world, and we need to really come up with a web of registries so that all of these are connected, such that all of these data sets are shareable and people are easy to share. Data is sharing between their different labs, and research groups are learning from each other. They are sharing their standards and so on and so forth. So we need a set of web registries, whereas all the data is accumulated in one place and it can be shared across the labs. But all of these solutions right now, if you think about all the publications in synthetic biology, either they are focused on E. coli or either the yeast. But we know from industrial biotechnology there are different organisms are used in industries, not only E. coli and yeast. There are a lot of organisms which have a huge potential for either it is a bioremediation or production of chemicals or antibiotics. So unless we develop tools for all of the microbes, I don't think so we'll be reaching a real potential of synthetic biology. I hope I have given you a brief overview of the challenges faced in at least some of the generalized way, and I enjoyed talking to you. Thank you.